Hi, I'm Nikki Alvarez, Associate Director of Alliances at Scripps Research in Calibr. And I'm really happy to be here today and share with you a snapshot of some of the ongoing translational efforts at Calibr directed towards new therapeutics for diabetes, and specifically type 1 diabetes. So type 1 diabetes is an autoimmune form of diabetes. So the cells that are targeted are beta cells, pancreatic beta cells that are responsible for producing insulin, which is responsible for maintaining glucose homeostasis. So it's an insulin-deficient diabetes. It's a growing problem, and it is an unmet need. So it's currently treated with subcutaneous injections of insulin to replace the missing peptide hormone, but this is imperfect and not a cure. So there's no disease-modifying treatments available. So it's recently, evidence has suggested that even though it's an autoimmune disease, that the beta cell itself is a friendly target. So it's enabled in sort of a self-destruction under conditions of stress. So in the context of diabetes, there's different stress pathways that can lead to programmed cell death. Cytokine stress or an inflammatory stress, something called ER stress, related to production of protein, and beta cells are particularly susceptible to these types of stresses because of the nature of the cell. Its primary function is to produce insulin, so it's a protein synthesis factory, basically. So what we wanted to do, and we partnered with JDRF to do this, but we wanted to identify therapeutics targeting the beta cell that could promote survival and ultimately perhaps delay or prevent disease, and then in combination with an immunotherapy, it could be a functional cure. So this program called the Translational Academic Research Partnership, or TARP, was an initiative designed to connect with leading labs around the world in this field. They may have a novel discovery that could use translation or a key resource of some sort. So this is a few examples of the collaborations that we've had within the partnership, all targeting beta cells in a different way, some phenotypic, some target-based, but all starting at the discovery stage of a screen. And they're at different stages of development today. But the first one I'll talk about is the most advanced, just in terms of the candidate status, getting to a candidate. So MST1 was discovered by Catherine Madler's group at University of Bremen to be upregulated in the context of diabetes and play a key role in beta cell death. So she had, well, just prior to publication of this discovery, we had partnered with her to try to target this therapeutically. But she published the story showing that, indeed, if you knock out MST1 in beta cells, that you lose, that you have protection in the context of a type 1 diabetic model. In this case, a beta cell ablated via streptozotocin, or STZ. So we did a target-based screen and identified, at the time it wasn't approved, it's now been approved, a known drug that had a novel activity on MST1. It's a drug for breast cancer, neratinib, known to target EGFR. So we subsequently profiled this in vitro and in vivo and showed that indeed this correlates to improved cell survival and function in beta cells. And then in animal models, the same animal model with the STZ, this pharmacologically recapitulated what she had shown with the genetic model. So from there, we set out on a focused accelerated medicinal chemistry effort. This successfully led to a novel series of molecules. And the one that's shown here is CAR432. This is a first-generation MST1 inhibitor where we were able to pretty dramatically reduce what is, in our case, an off-target effect, this EGFR, and increase the potency on MST1, and correlated that in vitro with beta cell survival in the context of various stressors, glucose-stimulated insulin secretion, and then on the right here, confirmation that this is also active in human islets. The translation to human can be challenging in this particular area, so that was important. So we have proof of concept in vivo as well, where you can see reduced glucose levels over time. And if you challenge the animals with a bolus of glucose, they have reduced curve. They don't go quite as high. They manage the challenge better. These are 2 different doses of CAR432, and we also see an increase in insulin both in the pancreas and the serum. So where we're at today is we're trying to select a candidate that we would take forward into formal preclinical development, IND enabling. So we have a few lead candidates where we've optimized a little bit more, or we've refined a little bit more the properties. We want this to be an oral drug and minimize other drug-drug interactions, et cetera. And we've managed to increase selectivity over MST2. So we have a few candidates now that we just need to put head-to-head in vivo and also assess some tolerability, toxicity, and we hope in the first half of this year to have a candidate. And then just real briefly, these are 2 other programs in the pipeline with JDRF. We have a program where we did phenotypic screens targeting mitochondrial apoptosis and cytokine stress. Interesting molecule that shown here in human islets does protect against cytokine-induced apoptosis. So we're currently evaluating this series in vivo. Likewise, for the UPR, we have 3 interesting scaffolds that have come out of this screen that was done to target the actual function of the ER, looking at reporters, and we've correlated that with various forms of protection in ER stress, glucolipotoxicity, et cetera. So I want to shift gears to another program in the last couple minutes, and so in addition to targeting the beta cells with small molecules and trying to find a functional cure, we have a program that is trying to create a next generation insulin that is liver biased. And this is a more physiologically relevant form of insulin. So the insulin that's injected today doesn't recapitulate endogenous insulin because it is injected peripherally. So it hits fat and muscle, other tissues equally or even to a greater degree than liver, and endogenous insulin is basically dumped on the liver, and that gradient is really important because the liver is a key glucoregulatory tissue, where it's the only tissue that can control glucose levels by storing the substrate. So we set out to do this, and you heard earlier today about Calibr's platform technology for antibody-based therapeutics. So we leverage that, in this case, making an antibody that targets an antigen that's only expressed on the surface of liver cells so that it will be biased to the liver. And we attached insulin to it and confirmed that maintained activity on the insulin receptor. And as a comparator, we made a point mutant that is similar size, similar pharmacokinetics, but it lacks the liver targeting entity by virtue of a single mutation that prevents it binding to antigen on the liver. So we've taken these molecules head-to-head. We've done a number of in vivo studies looking at biodistribution. So the accumulation of the liver-targeted insulin is greater in blue relative to the non-LTI in liver, and then you see that the bias is there for the liver, and there's more or less similar or less accumulation in fat and muscle. And you see the correlate of phospho-AKT, which is in the insulin signaling pathway, so indicating engagement of insulin receptor and activity in these tissues. And it controls whole body glucose homeostasis as insulins do. This is a very complicated metabolic experiment distilled into a simple graph just to show you that we have done what's considered the gold standard in this field for insulins. And in order to be able to say something's acting preferentially on the liver, this experiment allows you to do that with tracers. You can see where the glucose is going into peripheral tissues or into the liver and having activity on the liver. And in the bottom right here is 2 concentrations of LTI, where on the x-axis is the liver effect, on the y-axis is peripheral effects, so we've biased effectively towards the liver. The line in the middle is the normal insulin injected peripherally, where you see pretty equivalent effects on the periphery and the liver. And then our non-LTI shows some peripheral bias. So there's a few reasons we think we have an advantage over other people working on liver-targeted insulins. And We're currently trying to evaluate chronic effects on glycogen and other things that would be relevant to why this would be beneficial to patients. So this is just a summary of what I've said. Today we have kind of a broad approach to diabetes, looking at small molecules, biologics, and next-gen insulins, as well as regenerative medicines to improve life of patients with type 1 diabetes. And then we have early-stage ideas for other things that we're contemplating as well. And this took a lot of people in this, working on this portfolio, past, present, biology, chemistry, pharmacology, a number of collaborators, our screening facility, and all of our colleagues at JDRF who's funded all of this work, and we work very closely with and are very appreciative of their support. So, thank you.